Thank you very much for the introduction, and thank you for the invitation today. I wouldn't confess that I have any more expertise than lots of other clinician scientists and many of you in the audience in asking questions and making hypotheses, but it is a topic that we tend to neglect, and I think it is a really helpful thing to go over. So hopefully there'll be something useful in here for various levels of experience. So as I can't claim to be a world authority on this, I thought I'd start off with getting some quotes from the big thinkers of our time or recent times about the importance of questions. And as you'll see here, the right question is already half the solution to a problem, from Carl Jung. And then Einstein, if I had an hour to solve a problem, I would spend the first 55 minutes determining the proper question to ask. So you can really get a sense there how important a question is to them. Clearly, some of this is maybe not so directly relevant to what we're doing. I think to apply Einstein's logic to many of the things that we do, if you wanted to run a 5-year randomized control trial, you'd have to start at the age of 55, assuming you started preparing immediately after you were born. So, I think it's more the principles here that are really important rather than sticking to the letter of this. So, of course, we can't ask Einstein and Jung what's a good question. So the next best thing, of course, is going and asking ChatGPT and seeing what it will come up with. It always surprises me, the kind of answers that it comes up with. It's always interesting. So not a bad answer here in terms of a question being something that needs to stimulate thought, open up a conversation, be clear, specific, and relevant. But perhaps missing many of the things that we're really going to need to touch on. So we're probably going to have to go a little further than this. Whenever I get onto ChatGPT, I always end up asking it more questions, and I think that it's always interesting to see what it comes up with. And even asking it something philosophical like, is this a good question, seems to get it talking in the first person and trying to get into a philosophical conversation. So it's always interesting to see what you come up with there. But I'm going to leave ChatGPT behind just for now and move on to a bit more of what's out there elsewhere. So many of you will be familiar with this already, but we often take the meaning of words for granted. So hypothesis is a Greek word split into two parts: hypo meaning under, thesis meaning placing. And the statue you see on the right is Plato. You can see the word recorded in his writing around 2,500 years ago. But of course, this is something that humans have been doing for far longer than this. And it's really about our approach to solving problems. And to turn this into a more understandable concept, it's a supposition or a proposed explanation made on the basis of limited evidence as a foundation for further investigation, or to put it more succinctly, it's your educated guess that you will need to test. Now, one thing that we often get wrong, or that you might see incorrect in, let's say, a grant application or a paper, is that people use these terms interchangeably, and that they're not. So they are very distinct things, a hypothesis and a question, but they are complementary, and you need both. So questions are the start of your process when you're making a decision. You're trying to decide what to do, and you ask yourself a question. But quite often you're going to encounter a scenario where there is not an answer available, or you're not satisfied with the information you have to produce that answer. And it's those difficult questions, those unanswered questions, that lead to your hypothesis. So you're coming up with an idea of what you predict is going to happen. So it's a statement, it's not a question. So if you see a hypothesis in a grant application ending with a question mark, that's not really a hypothesis. And it might be splitting hairs, but as you'll see when we go through, it's really important that you get both of these elements correct, because it's fundamentally important for the rest of your research. So in the end, your hypothesis is going to frame your approach to answering the question. Now, this might all seem incredibly obvious, but this analysis of randomized control trials, so you think, at the pinnacle of clinical research, published in two of the sort of highest ranking clinical research journals in 2015, and they analyzed every single randomized control trial in that year. And what they found is that approximately a quarter of papers had no clearly stated hypothesis. And you might imagine that this is something that's just kind of implicit in there, that you'll be able to kind of establish it from reading the introduction, but really it's a a fundamental part of research and should be stated in here. It's the basis of everything that we do. So it's disappointing that actually it doesn't seem obvious, or that people take it for granted. And I'll show you some of the reasons why this might be later on. So, as I said, your hypothesis is the basis, or hypotheses are the basis of all science. But it's important to think about them in their wider context, and they're really part of a cycle of research. So you're forming your question and your hypothesis, your linked hypothesis, using what existing knowledge there may be. And then you will be able to conduct some experiments, some research to test your hypothesis. And then you're going to get that peer reviewed, and hopefully you've answered the question robustly. You've come up with a clear answer that your peer reviewers are happy with, and then you can publish it, and it forms part of the existing knowledge. And that then feeds into your own questions and hypotheses for the future and the wider scientific community. And this is something that is done at all kinds of different levels. And we often get focused in on a hypothesis for a paper, for example, but this goes right as high as, you know, a hypothesis that might define an entire career. And so you could take the example today of it was the announcement of the Nobel prizes for chemistry today. One of the winners was called Demis Hassabis. He's a co-founder of Google DeepMind, and he, together with colleagues, hypothesized a long time ago that it would be possible to use artificial intelligence to predict the structure, the three-dimensional structure of proteins with just the amino acid sequence. And that is something that no one had ever been able to get close to achieving before, and required lots of complex laboratory assessments to come up with a direct evidence instead of a prediction. And obviously to win the prize now, so he has convincingly taken forward that hypothesis, but as part of a journey over many, many years, over a career. But you can take this right to the other level where, let's say you're a laboratory scientist and you're doing an experiment, maybe producing some pilot data. You might be able to form a hypothesis and answer that within a day. And of course, in between, you've got the work in a paper where you would generally expect to state a hypothesis at the end of your introduction, and in a grant application, which is an even larger piece of work. And grant applications are always framed around an original research hypothesis. So these are things that you're going to be thinking about at lots of different levels, much of the time. So this all starts with the question. So what makes a good question? And we found before that it was not possible to get that kind of answer from ChatGPT. Well, I think in terms of what we're thinking about, good questions will lead to important answers. And what's even more important is that whatever the outcome you get when you ask the question and test your hypothesis, the outcome, even if you disprove what you hypothesize, will still be important. If you can find a question like that, then you know that as long as you do your study well and you address your hypothesis properly, it doesn't matter what happens, you're going to find something important that's going to change what people think and change what people do. So if you ask a good question and you conclusively answer it, you're doing really good science. So just to give an example here, this is some NIHR funded research and involving some people in Leeds, but lots elsewhere too. And they hypothesized that opening up narrowed coronary arteries, heart arteries, that were limiting blood supply to the heart could improve long-term survival and improve heart pumping strength in people who've got weakened heart failure. And that seemed like a fairly it's a very credible hypothesis. And many people, I think, expected this to be a positive trial in inverted commas, but what they found is it made absolutely no difference. And on one hand, that's disappointing that we haven't come up with a new treatment to help outcomes and to improve heart pumping strength. But on the other hand, there's no uncertainty about this now. We know that this is something that doesn't help patients, and so we shouldn't expose them to that risk because it doesn't provide any benefit for them. So in spite of their hypothesis being disproven, this is practice changing and will make a big difference to patients, and that's what we need to be achieving. So how do you start making your own research questions? Well, it's a difficult thing to do, and it relies expertise from you and the wider literature and anyone else you can get it from, really. And what you're trying to do is to establish what are the big gaps in knowledge or practice in your field? And that might be a difficult thing to appreciate when you begin, and you might need some help there, and that's absolutely fine. Often it's helpful to think about when you're doing your day-to-day job, assuming you're in a clinical job and you're trying to help a patient to make an informed decision about something, where do you struggle to help them to make those informed decisions? So what information are you lacking? What questions are there? What important questions are there not answers to? And that's going to be a really useful guide. Of course, it's important that you know what's already out there in the existing evidence, but also that you don't take for granted that things that are out there and, in inverted commas, known are actually robust. So you need to question what's out there as well, to question the validity of it. And in the end, if you think you've got a good question, you need to kind of take it forward and imagine, well, if I answer this, and depending on the different outcomes, what would be the impact of clear-cut data on clinical practice? And that will really help you to come up with in your own mind about whether this could be important. So I mentioned that this is something that can come from you and maybe from colleagues. Of course, there's lots of other lots of other ways that you need to think about this as well. So one might be that you could look at sort of clinical knowledge that's already been synthesized and needs have been synthesized by other organizations. So these might be clinical practice guidelines, NICE, government. You keep on thinking of lots of other organizations that can do this. Of course, it's really important, as I'll talk about in a minute, for patient and public involvement in this process too. So that is a fundamental requirement when you're coming up with a research question and hypothesis. And you may well need to do something more objective and try to bring together all of the relevant evidence in a systematic review and potentially even meta-analyze it to do a sort of a statistical analysis of all of it together, so that you can come up with the best possible idea of where the knowledge gaps are and where you can make a difference. So where does PPIE fit in? Well, you see from the figure that I've taken from another website here that, of course, it's relevant throughout the research cycle, but obviously we're focusing in on hypothesis generation and the question that's driving that hypothesis today. So you're going to come up with questions, but you obviously are going to be needing to involve PPIE groups in this process as well, because the questions that you come up with might not be the same as the questions that they come up with, and you need to find questions that are important to patients and to the public as well. That's because obviously we need to be coming up with answers to questions that are important to them, and generally speaking, public money's going into this, so we need to involve all of the kind of stakeholders in this process. And then when you've got your question and you form a hypothesis, you need to make sure that you're going to be able to test this hypothesis in a way that people are going to want to participate in, because you might have a really interesting question and hypothesis and people might be able to agree, including in a PPIE group. But if you can't come up with a way that you can really test that hypothesis in an acceptable way, then it's still really not going to be successful. So it's fundamentally important that you have PPIE involvement. So obviously it's difficult to do something interactive here, so I apologize about this. So I'm going to have to just go through this for you, but I was hoping if it was a slightly smaller group that we might be able to get some answers as we go along here. But some examples of research questions. There's 4 here, and one of these you may have heard of Ig Nobel Prizes. So these are prizes that are, if you like, the joke version of the Nobel Prize, awarded to really kind of strange, unusual questions and projects that people perform, but perhaps with an interesting message underlying it, even though it's not really a very good research project. So if I run through them, does dexamethasone improve survival in people hospitalized with severe COVID-19? You may well be familiar with that question. That was actually the basis of part of the recovery trial. So in the end, they tested that question and found that a cheap medication could reduce all-cause mortality by over 1/3. So that was really practice changing across the world, really important question. So clearly that's not the Ig Nobel Prize winner. The next one you're probably aware is going to be the prize winner. So can poorly controlled asthma be improved with roller coaster rides? Well, I think it's sort of self-evident that you're not going to be able to improve asthma control by going on a roller coaster. And we'll talk about how thinking around a few properties of a question might help you to sort of decide early on that that would not be the good kind of question to ask. The reason that won the prize is they showed that people's asthma did in fact appear to be better controlled, and they speculated that that was because it changed their perception of breathlessness. Now, I haven't read the paper, so I'm relying on the Ig Nobel Prize Committee for that, but I think it makes the point that it's probably not a very good question. Is smoking a cause of lung cancer? Well, we know now that it is, but that was once an incredibly important question to ask, and it has made a fundamental difference to the way that we approach life now. And then last of all, one of the sort of recurring questions in my field of research, does chocolate reduce cardiovascular risk? Not really a very imaginative question, not really a very good question, really. It's kind of saying, well, I like chocolate and I don't like heart attacks, so maybe I can put both of those into one hypothesis or one question, and wouldn't that be great if I could eat chocolate and reduce my risk of a heart attack? Which, in general terms, is not what we think. So how can we think about these things? Well, I think one approach that I think is useful is to try to look at, first of all, we've said that you need to find questions that are impactful. And so up here in this graph, and if you have a potential impact, if you prove your hypothesis, is it going to be impactful? Then that's great. But you also need to have some kind of, it needs to be likely that your hypothesis is going to be proven correct, that it's credible. So in the case of, let's say, if I said, I hypothesize that putting a stent into a narrowed heart artery reduces your chance of dying from bowel cancer. Well, that's just completely implausible. And so even if you were to prove that if you were to prove your hypothesis, then people wouldn't believe that, because the likelihood of that being true before your trial was really not strong, and there's no credible reason why that would happen. So you're probably not going to persuade people with that. And if you disprove your question, people are going to say, well, of course, putting a stent into a narrowed heart artery doesn't reduce your risk of dying from bowel cancer. So you need to have a credible kind of hypothesis that you raise as a result of your question, as well as having impact that comes from that. And I think this is a helpful way of thinking through and making sure that you are going to come up with a question that has impact and is going to genuinely have the potential to change what people do and what people think. So if you've got a question and you want to turn it into a hypothesis, how do you go about that? Well, I think, as we've seen already, good questions are quite often open and ambiguous, and hypotheses are really the opposite of that. So they're very narrowly defined and unambiguous. So that means that you can often end up with multiple different hypotheses that could come from the same question. And the most obvious version of that is that you could hypothesize that, let's say, the outcome improves with an intervention, or you could hypothesize that the outcome is unchanged with the very same intervention, or you could hypothesize that the outcome is worse with that intervention. So there are three very kind of obvious examples of how you could end up with different hypotheses. But in fact, there's a lot more nuance, as you'll see when we come through, in how you frame a hypothesis and the ongoing way in which you test that. So there's still quite a lot of ambiguity left in hypotheses, but you're trying to remove some of that as you go through this process. So if we follow through this kind of pathway, you're starting with your good question. You need your hypothesis to remain relevant to that question, obviously. You need to try to come up with a link between a dependent and an independent variable. So using the example I've been giving, the link between stenting a narrowed heart artery and survival in people who've got weakened heart pumping. So we're making a clear link between a dependent and an independent variable. We need to state that in a specific and unambiguous way. So we need to be really clear about what we think is going to happen and how that's going to be defined. And it needs to be testable, such that you could lead to a true or a false outcome. And the point to really make there is that if you come up with a hypothesis and then a design to test that. And it's only possible for you to reveal an answer that is either proving your hypothesis or inconclusive. So it's basically impossible to come up with a disproof of your hypothesis. Then you've basically failed in producing a hypothesis, because you need to have all possible outcomes for you to be able to adequately test this and come up with something that is going to change what you do afterwards. You might be thinking at this stage, well, do I have to have a hypothesis? And you don't, but it's really important to think about the implications of that for your work. So sometimes there might just not be enough information out there for you to make a hypothesis, to make an educated guess. And in that case, it's perfectly reasonable to do some research that produces the information that will help you to make a good hypothesis. And you might hear about this in the term hypothesis generating research, and that's absolutely fine. But what you mustn't do, and unfortunately what a lot of people do, is that they do this and then they make conclusions from it. And of course, they haven't made a hypothesis before they've done their work. They're making a hypothesis at the end. And that's then completely unfair, because the whole process has been biased. You need to set your hypothesis before your experiment so that it's clear that that was not influenced by what you know during the study. So by all means, you can do this, but you have to be aware that the data you produce at the end is not going to be useful to change what you do, only to make hypotheses. So I'm going to start with a few examples of a question and a paired hypothesis and just think through these. So the first question is shift working bad for your health? And you could very easily hypothesize from that, yes, shift working is bad for your health. So I'm sure you're thinking already about some problems with this, and I'm afraid as we haven't really got a very interactive way of being able to do this, I'm just going to have to talk through it. And the big problem there is that it's incredibly ambiguous. So what does bad for your health mean? What does shift working mean? You know, if you work an evening shift, does that count, or do you have to be working night shifts? How often do you have to be working night shifts? So you know, there's a lot that you need to define there, and if you sort of approach it without that information, you can't really test this. So perhaps you could improve this by saying people who work night shifts have higher blood pressure than age-sex match controls working only day shifts. And you can see that that's a lot longer and wordier. It could include even more detail. So I haven't sort of specified, do people need to work only night shifts, or could they work a mixture of shifts? And so you can get the sense that sometimes you could take it to a level where it's no longer really a very useful hypothesis, and perhaps you're just going to have to sort of narrow down some of these details somewhere else. And so there isn't a right or a wrong answer here. Sometimes it's better to have a slightly more brief hypothesis, but clarify and pin down some of the details in your experimental design. And as long as that's all fixed before you start your study, then the key things are in place there, and that's absolutely not a problem at all. So the next example is the COVID-19 vaccine a cause of stroke, and so you could just flip that around again and say yes, that vaccine is or can cause stroke. And again, I'm sure you can see the same kinds of limitations here, that this is really very ambiguous. So, you know, lots of things you could sort of take the vaccine out there and swap it for something else, like insect bites or all kinds of other things, and maybe you could produce some kind of tenuous connection between them there, but it's not really a testable statement at the moment. So you need to pin this down more. So in one way, you could, for example, say in a randomized control trial, people allocated the vaccine experience a higher rate of stroke than those in the placebo group. And of course, those of you who have more familiarity with this might sort of recognize that, well, of course, this depends on the size of the trial, what kind of rate of stroke you could detect. And of course, there are all of these kind of statistical considerations and things as well. But I'm just trying to make the point here that this is, you need to kind of pin down precisely what it is that you're proposing before you can begin to test it. So you need to have a more tangible idea of what it is you're proposing is happening. So the last example I'm going to give is, are 3 days of antibiotics as good as the traditional 5 days for treating pneumonia? And turning that into the hypothesis that antibiotics given for 3 days are as effective as for 5 days in the treatment of pneumonia. So again, it's a bit of a recurring theme in here, that it all comes down to ambiguity, and that ambiguity affecting our ability to test this. So we haven't really said, we haven't said which antibiotics, but perhaps that's probably less important. But in particular, we haven't said what as effective as means. And so we need to pin that down some more before we can test it. So you might say the antibiotic treatment for 3 days leads to similar treatment failure rates. But again, that is still a bit vague, really. What does treatment failure mean? So in this case, I've changed that to say antibiotic treatment of pneumonia over 3 days is non-inferior to treatment over 5 days, as defined by the 30-day rate of pneumonia hospitalization. And I could put and death in there as well. Well, but you get the sense that this is more clearly defined, and I've actually said, rather than as effective as, non-inferior, which is a bit more of a technical way of describing the level of the margin of difference that we might be able to tolerate between the two, which we don't really need to worry about for now. So those are just some examples. Obviously, in your own setting, you need to kind of go through this with other people and think, well, what's right for you. You can, of course, ask colleagues. We've discussed that going to PPI is really important, and it takes a lot of time to get this right and to fine tune it. I was, of course, on ChatGPT before, and I thought it'd be interesting to see what it thinks of the chocolate reducing cardiovascular risk hypothesis. And actually, it doesn't do a bad job of coming up with some ideas again about how we could pin this down. So it makes the suggestion that we need to be more specific again. So maybe discussing what kind of chocolate, what kind of people. We need to really say reduces cardiovascular risk. Well, what does that mean? Is it your blood pressure or your cholesterol level or your development of heart attacks, for example? And you may well need to sort of pin that down even more, so specify some levels of those factors so that it becomes testable. So I wouldn't recommend that you go and test your hypotheses, or to ask ChatGPT about the quality of your hypotheses. I would use all of the other resources you have instead, but it doesn't do a bad job. So let's say the hypothesis is ready now. So somebody's put it into a grant application. So it's gone into an advanced fellowship application, and they're asking the question that we were there before, and the hypothesis that we came up with about 3 days of antibiotic treatment being non-inferior to 5 days of treatment. So we can say it's an impactful question. You know, it could lead to reduced rates of antimicrobial resistance and all kinds of other beneficial factors coming from it. We can assume that maybe they've got some good pilot data showing that this is a reasonable presumption to make, and they stated a clear hypothesis. But of course, in a grant application, there's a lot more in there. So we need to think about, well, what else do they need to know? And this is the other kind of really important part of this. It's great to have an important question and a good hypothesis coming from that, but you need to be able to test that hypothesis. And so, you know, we need to think about a lot more about how realistic that is. So what would the design of your study look like? And this is a really complicated thing potentially for this study that we're talking about. So it might need it's going to be a randomized controlled trial. It's quite possibly going to need to be multi-center. And, you know, you might be a domain expert there, but you're almost certainly going to need expertise from people who are specialists in clinical trial design and conduct. And you're going to need to think about, are you going to bring together everything that you need to answer this, address this hypothesis? It can be a really brilliant hypothesis, but if you can't address it, then it's not the right one for you. So have you been through it with your PPIE group? Are you going to be able to recruit enough people within the where you're performing the research. Are you going to have the right research team to do this and all the other sort of physical infrastructure you need? And are you going to be able to bring in the money that you need from a funder like NIHR to do this? One thing that I haven't touched on too much because it's not really the topic here, but with things, we can have quite a well-defined hypothesis, but there can still be a lot of ambiguity in there. And in this example, we talked about non-inferiority, which basically it's a description of we have to specify the level of tolerance we have for two things being close to one another, such that we assume, well, it is probably broadly the same as and not worse than. And you can specify different levels where you begin to say, well, it's gone from being probably the same to, or in other words, not worse than to worse than. And there's not a right or a wrong answer there. So all of this can then determine how large a study is and how much it's going to cost and how feasible it is. So this is why you need to get experts in for all of these stages and think really carefully about your analysis plan. So effectively, what all this boils down to is, can you deliver on testing the hypothesis here? Because if you can't, then you need to find a different hypothesis. So assuming that you get your work funded and you're ready to start using your hypothesis and design, before you start conducting a study, it's good practice, and in fact, generally speaking, mandated a lot of the time for you to submit your plan to somewhere like these websites that I've listed here, so that they're there, publicly accessible, and that when you finish your study, people will be able to go back and make sure that what you've done is what you said you were going to do. Because of course, there's always examples of where trials have changed in their design because it looks like they're not going to fulfill the requirements of the trial. They're not going to lead to a proven hypothesis. Who knows the motivation in every case, but certainly trial designs are known to change, and this is why we do this, to try to stop that from happening, because it's not really then properly testing a hypothesis. So strangely enough, on these websites, they don't actually have a box that gets you to list a hypothesis, even though many of the studies that go on to publish and refer back to these websites will state a hypothesis in their paper. And that does seem slightly strange, but usually speaking, when you go through the trial design, they will match one another very closely, or hopefully entirely. And that's a really important thing to check. So it's a really good thing for you to do, to make sure that you've proven to everyone that your hypothesis and your plan was formulated before your study. And this, not only helps you to show that you did this really well and fulfill your commitment to open research and all of this kind of thing, but also to help others to, maybe other funders, to avoid funding an overlapping study if that's a part of their strategy, not to sort of fund overlapping trials. Maybe it will help other people to help you recruit into your trials. So there are lots of potential benefits from this. So then we get to the stage where you've tested your hypothesis, and you may well wonder, can you put your feet up now? You've tested your hypothesis. Is your job done? Well, as we talked about before, this is a cycle, so no. And so, in the various scenarios, if we start with, let's say you've proven your hypothesis, well, that's great, but I put it in inverted commas there because, not everyone might agree that you've proven your hypothesis, or it may be that more data emerge in future, and that brings into question the totality of the evidence may be more equivocal then. And so, we shouldn't ever sort of assume that things are proven altogether. But the same applies for when things are disproven. Often people disagree there, and maybe rightly or wrongly. So I give the example of that trial I showed at the beginning of stenting narrowed coronary arteries in people with heart failure, which didn't show a benefit. There are some people out there who think, or maybe there's a group of higher risk patients who would still benefit from this and think that other trials would be justified, and it would be up to them to be able to persuade other people of that equipoise to justify ethically a trial and to get the funding for it and so on. But this is what happens, that sometimes the answers are not always clear cut, or that some people feel that there is still sort of narrow questions that remain to be sort of defined and tested. And quite often we're in a position where studies just don't properly answer a question, and for all kinds of reasons, and that uncertainty is left, and then it may well be that the same old hypothesis remains there and still needs to be tested. So you need to think about all of these things carefully when you get to the end of your study. So we're on to the wrap-up stage now. I think we're doing all right for time. So I think the key take-home points here, just to whiz through them. Hopefully, you knew this already, but hypotheses are the basis of science, and we've been through how questions lead to hypotheses, which lead to study designs. And so what you need to be aiming for is to come up with a good question, which means that it should lead to an important answer, irrespective of what you find. Your question you're going to turn into a good hypothesis by making sure it's clear and testable. And you need to make sure that you robustly test that hypothesis with a good design to lead to impacting clinical practice for patient benefit. This is something that you need to spend a lot of time thinking about before you're actually getting into testing your hypothesis. So don't worry if it's taking you quite some time, potentially years to get this right, because it's going to save you time getting it right, rather than spending 5 years running a trial that was, it started off on the wrong footing. And we've also discussed the importance of involving patients and the public throughout this process. And in the end, as you've seen, this is going to be a process that you do throughout your career. So even when you've answered your current hypothesis, there'll be plenty more for you to work on. So I'm going to wrap it up there. I'll probably stop sharing my screen so I can see what's going on. And yeah, so I think we have about 15 minutes left. Is that right, Yus?
Yes. Yeah, so thank you, Rick, for enlightening us about how to develop good research questions and hypotheses. So we've got one question so far. I think it's a little bit technical because we do have people of various backgrounds. So I think maybe they just need a little bit of guidance of it. So I think the question asking if we want to know the effectiveness of the interventions for tuberculosis stigma to inform new and culturally adopted interventions, I think they were asking like, what do you think could be the right thing to do in terms of hypothesis and also research question to separate that? I think they're quite technical, this one, isn't it?
Yeah, I have to say it's not something that I would claim any expertise about. So I wouldn't want to wade in there and get it wrong. But I think you need to sort of use the knowledge that's out there. So it might involve performing a literature review, talking to other experts in the field, to patients and the public, and bringing that all together and trying to identify what's known, what's not known, and then that can help you come up with thinking about what would be an impactful hypothesis to test. But I wouldn't really want to go any further and give specifics, if that's all right.
Yeah, great. Thank you, Rick. So we've got Joanna Slater.
Hi, I might be one of a few that are doing the health research at Leeds Beckett. So I'm starting a dissertation and we have two options. We can do secondary research, systemic literature review, or we can do a work-based research. And we had our initial tutorial and it was really focused on everybody doing systemic research, but because I'm an anomaly, I thought, no, I want to do the work-based research. So I just wondered, because I didn't really get to grips with understanding what the work-based research looked like, would that need a hypothesis?
I think if you're going to be performing primary
any kind of research you would do
then yes, I think you will need to, and of course all the other things that I kind of glossed over, like ethical approvals and so on, potentially depending on your research as well and so on. But yes, in order for it to be, well, I suppose the only other approach I would say is I touched on the hypothesis generating side of things. So if there were no information out there that would allow you to make a good informed hypothesis, then it would be justifiable to do the hypothesis generating research, which is really collecting data which you hope will be able to help you or other people make a hypothesis later on. Of course, I don't know how much time you've got to do your project, but it all needs to be realistic within that period as well. So there's lots of things that need to be weighed up to make sure that you can deliver on the design, whether you're testing a hypothesis or not.
Thank you. Thanks for answering that. I kind of thought so, but I just wanted clarity. Thank you.
Yeah, no problem.
Yep, so just got a few questions, Rick. So it's good that you mentioned that we have to set the hypothesis as priori rather than getting your results and then come back. But sometimes it's not quite clear. Like when you read papers, sometimes people are actually not really clear in stating what their hypothesis and potentially even objective even. So do you have any advice, and what we should do, for us to be transparent?
I think it comes back in an ideal world to making sure that you place in a public repository your hypothesis and your research design in advance. And that's, as I said, it's mandated, generally speaking, for randomized controlled trials, but you can do it for other trials as well. But as I also said, the way that those websites and repositories work, they don't always encourage you to list things like a hypothesis. So I think then it's the reliance is on the researcher to make sure that they include some of those things. And it's frustrating doing that in some ways. It's something extra to do, but it really does help you to demonstrate to peer reviewers and journals and to the world afterwards that you haven't done that you've actually really clearly set out with a plan and you stuck to it, which means that the answer is much more compelling than if people suspect that maybe things have moved during the process.
Also, I think we've not spoken because the title is about research question and hypothesis, and we've got quite a lot of people backgrounds here, from people who wanting to do research, some PhD students, and us as well, actually as supervisors here. Sometimes usually the tricky bits in writing thesis or report is to separate or distinguish between the aims and objectives. Do you have any insights or tips, to help everyone else here?
Yeah, I mean, I think again there can be some kind of overlap between aims and objectives. So I wouldn't sort of necessarily say that they're particularly different from one another, but a hypothesis, it tends to be a kind of, as we say, an overarching kind of sort of testable statement. But you might, for example, in a randomized control trial, you might have a primary outcome measure, and you might have a variety of secondary outcome measures that you want to pre-specify. And so those could be your aims or your objectives. So you could list those and say, we want to test all-cause mortality after 3 years for our primary outcome measure, but for our secondary outcome measure, we want to look at quality of life, or the change in quality of life after 3 years, and etc., etc., etc. Or for whatever study design you're working with. So it's really kind of breaking down, giving a bit more detail to what you're doing. But I don't think there is a hard and a fast rule about how you do that. I think the key thing is about it's really the hypothesis, which is the thing that is really needs to be set in stone before you start, and then the design around that. And you can break that up into objectives, or you can leave it in text or whatever you like. The main thing is that all of the information is there, so that you set out knowing exactly what you're going to do and why, and that doesn't change.
Okay, so we probably have one last question here. So by Priscilla. So she's looking at education and training of healthcare professionals in pharmacogenomics. So her question is, how do I engage patients or the public in developing my research questions, as current literature shows that most people don't know much about what pharmacogenomics is?
Yeah, so that's a good question. I think many of the organizations that people attending will be part of may well have PPIE groups. Sometimes those will be quite broad. Sometimes they will be very focused around a particular disease, let's say, or a particular group of people. So yes, what you could do if you don't have a pharmacogenomics focused group is to see whether there are other centres in the UK that do, and often they're very happy to collaborate and help if they recognise that they're doing something unique in the UK or fairly unique. Otherwise, maybe organisations that advocate for pharmacogenomics and pharmacogenomics research in the UK or globally might have PPIE groups as well. So that it doesn't necessarily need to come through your own organisation or another university or NHS organisation. There may be other bodies. I suspect not for pharmacogenomics, but sometimes there are patient groups as well that you can go to directly. So there are lots of ways of doing this, and I'm afraid I just don't have enough awareness of that field. But if you talk to other experts in the field, I'm sure those people who are designing pharmacogenomics research will be able to direct you to appropriate PPIE groups.
And just to touch on that, we also have a seminar series on embedding PPIE and also EDI in research. So that is planned for 29th January next year. So hopefully you can get more information from that as well. So I think that's all we've got time for now. So I'd just like to thank Richard for delivering your talk today. And just to let everyone know that from tomorrow, we will send everyone's a link for a feedback, and we really value your feedback so that we can improve our session, and also what are the topics that you want to appear in the seminar series. Also, once you click the feedback survey, then you will also get a certificate so that you can put that on your, on your portfolios. And just letting you know, I think there's some thing talking in the chat as well that the next series will be on the 6th of November. So here we will have Professor Anne-Marie Keenan will be talking about developing a personal and professional training plan. And also 2 weeks after that, we will have Professor Anne Morgan and Professor Hemant Pandit, which will be discussing pertaining to mentors, supervisors, and research advocates. And after that, we have a break, for Christmas and New Year, and we'll continue after that. So yeah, so just stay tuned. The advertisement will come out shortly. So thank you everyone, and have a nice day.